Hello, everyone. My name is Randy Struby. I'm the Director of Global Marketing at InSphero, and I'd like to welcome you to today's webinar, Proteomic Profiling of 3D InSight Human Liver Microtissues with HRM-MS. Today's webinar is being co-hosted by Biognosys AG, a valued partner of InSphero, who also happens to be a neighbor of ours in Rinner. And today we'll be covering some recent studies characterizing InSphero 3D liver microtissues using an advanced mass spec technology from Biognosys called hyperreaction monitoring mass spec. And we appreciate you taking the time to join us today, whether it's late in the afternoon or early in the morning. It's my pleasure today to introduce today's speakers, Katrin Roessger from InSphero and Dr. Oliver Rinner from Biognosys. Katrin has been an application scientist at InSphero for just about a year now and is part of the product development team focusing on the investigation of novel liver microtissue models as well as optimization of assays and protocols for use with these organotypic 3D model systems. Katrin will be giving us a brief introduction to InSphero's 3D human liver microtissues, and these were the models used in our study with Biognosys. Following Katrin's introduction, we'll be joined by Dr. Oliver Rinner of Biognosys. Oliver graduated at the University of Tübingen in biochemistry and psychology, and then received his PhD in 2005 for his work in the field of psychophysics and molecular genetics from the University of Zurich. He did a postdoc in the lab of Dr. Rudi Ebersold in the ETH Zurich, where he published key papers and patents in the field of targeted proteomics. He then went on to found Biognosys in 2008, and he'll be giving us a more in-depth look at the Biognosys next generation HRM-MS technology and its applicability to 3D microtissues, and this will include a review of some recent data that was published in collaboration with InSphero on proteomic profile changes in acetaminophen-treated liver microtissues. And this was published this past February in the journal Molecular and Cellular Proteomics. So with that, I will hand the stage over to Katrin. Are you there?
Yes, I'm there.
All right. You should have control, and I will hand it over to you. Thanks.
Okay. Welcome, everybody. Today I want to give you an overview about our 3D liver microtissues and how we actually explain to you a little bit more how we at InSphero use and in the end also generate and use them, And so you know that actually our main focus is liver at InSphero, and the reason for this is that the liver plays a central role within the body in transforming and catalyzing and clearing chemicals, and it is also susceptible to the toxicity of different agents. So for example, if someone is taking up medical agents in overdoses or sometimes even within the therapeutic range, those agents might cause liver injuries, and due to this reason, many drugs have been withdrawn from the market over the past years. And this hepatotoxicity also accounts for a substantial number of compound failure during the clinical trials. So this is actually highlighting the need of new and good drug screening assays that you can kick out non-potential compounds already before you even go to the clinical trials, and that you thereby can save time and money. As you can see, the liver represents a complex network and it contains different cell types. Besides the hepatocytes, we also have Kupffer cells, stellate cells, and endothelial cells, and many more. And our goal at InSphero is actually to combine all these different cell types in order to create a spheroid model which is as close as possible to the native biology. A good example on how we did and reached this goal so far is shown in those two pictures, which were kindly provided by Professor Hengstler. You can see confocal microscopy of, on the right, the native human liver, and on the left-hand side, the human liver microtissues we created. And this is a staining, a DPP4 staining of the bile canaliculi network. And what you can see is actually the similarities between the two pictures. You can see in green the bile canaliculi, and it's in the human liver connected with each other, as well as shows blunt ends, and this is what you can actually also see in our human liver microtissues. So we are already really close to make it to native biology. I will shortly introduce you to our microtissue production procedure. So we are seeding our cells in a hanging drop format in our microtissue production platform, which we call Gravity Plus Plate at InSphero. And after a maturation period of 2 to 4 days, where the single cells agglomerate together and transform and form a microtissue, we transfer our microtissues by adding an additional media drop. We are transferring them to the microtissue culture and assay platform, which we call Gravity Trap Plate. And when these spheres are in this trap plate, we can further run our experiments. On the left-hand side, you can see how the spheroid, the microtissue, looks within the gravity trap plate. And usually we are producing microtissues of a size of about 250 to 300 micrometers, but depending on your project and depending on your wish and aim, we can either decrease or increase the size of the microtissues. The reason why we are going for 3D instead of 2D is easy, because two-dimensional tissue models totally lack of complex structures and realistic complexity. So even if they are amenable to high throughput screening and are capable of detecting metabolite-driven toxicities, they have several disadvantages. Besides the poor negative predictive value, the major drawbacks are the loss of liver-like phenotype during the culture, and with those 2D models, you're not able to do repeat dose studies, which is actually very important if you want to test your drugs. The 3D microtissues, on the other hand, mimic the native biology more accurately by capturing a greater phenotypic heterogeneity. We also have changes in gene expression and cell behavior, so this is actually pretty close to nature. The 3D microtissues are more assay flexible, so we can cultivate our systems microtissues for up to 28 days. When they are still being stable with their conditions, and with this 3D microtissues we are able to do repeat dose studies. At InSphero we are able to create liver microtissues from different species. So besides human and rat, we can also generate microtissues from mini pig, dog and monkey, as well as from the cell line HepRG. And we are even able to co-culture them with the NPCs, so the Kupfer cells, stellate cells, the ones I mentioned before. And this is also upon request, depending on your model. This actually makes the 3D liver microtissues available for many applications. So besides the measurement of acute toxicity or mitochondrial toxicity, we can also test your drugs if they maybe induce oxidative stress or lipidosis or cholestasis. We can also carry out some drug-drug interaction tests for you or other DMPK studies. And we are also now focusing on modeling liver diseases, such as fibrosis or steatosis. So, for example, if you're interested in a fibrosis model, then we can, beside the hepatocytes, we can seed Kupfer and stellate cells and let them grow with each other. Or maybe you have developed an antibody drug conjugate you want to have tested, then we can combine the hepatocytes with Kupfer cells and endothelial cells and thereby generate a personalized model for you. Furthermore, we are able to connect different species with each other, and we are able to do a single cell type or multiple cell type co-culture setup. Those various applications also make the give us many endpoint assays we can measure. So beside our standard, the ATP albumin secretion or the CYP activation, as well as the Kupffer cell function tests of IL-6 secretion or TNF-alpha secretion, we are also able now to measure transcriptomics and proteomics. And this proteomics analysis is actually carried out by Oliver and his team, and I'm pretty sure he will explain it in more detail to you in the second half of this webinar. Furthermore, we are now focusing on the measurement of mitochondrial toxicity by using the Seahorse machine, and this is a service we can hopefully offer to you within the next month. In the last couple of minutes, I would like to focus on a human liver microtissue study we did recently, where we tested 3 donors derived from cryopreserved primary human hepatocytes in co-culture with primary NPCs. So how did we do that? So we generated our human liver microtissues as the production procedure I've shown before. So we are seeding our cells in the gravity plus plate and then transferring the microtissues to the gravity trap plate. And we are culturing our human liver microtissues for 35 days. And every 7th day, we are measuring different parameters, which is the ATP, the albumin, then we're taking histology samples, we measure the CYP activation, proteomics, and transcriptomics. And here in this short study, I will just focus on the size, the ATP, albumin, and CYP activation. The proteomics is later on shown by Oliver. So coming to the results, so what we actually obtained was we were able to reach a stable microtissue size of all 3 donors over the full production cultivation, up to the 35 days. Furthermore, we had a preserved viability over the full length, and which is indicated by ATP, and we also had a stable functionality, which is illustrated by the albumin secretion for all 3 donors, and the albumin secretion for at least up to 4 weeks. We also did some CYP activity measurements. So in this case, I will just focus on 2 major cytochromes which are involved in drug metabolism, and they are catalyzing different reactions in the drug metabolism. One is the CYP1A2, and the other one is the CYP3A4. And when you have a look on the different donors, you can see that they have a different expression profile and activation profile of the cytochromes. So if, for example, for the CYP1A2, you would say that donor 3 performs best, whereas for the CYP3A4, actually the donor 1 seems to be the best or most attractive. So depending on your drug and which pathway it is influencing within the liver, you can choose between several donors and actually in the end pick the best one, which in the end fits best to your purpose. So to sum up, we can conclude that the 3D human liver microtissues we created were stable, showed a stable viability for more than 4 weeks. We had a stable functionality, retained albumin secretion, and retained CYP activity. We also had a constant phenotype, so a preserved liver-specific gene expression, and we were able to do a co-culture with the Kupffer cells. And this, in the end, ideally suits our liver microtissues for various applications, the ones I mentioned before. So with this, I'd like to thank everyone, and now I will hand over to Oliver.
So welcome everybody. My name is Oliver Rinner from Biognosys, neighbors of InSphero, and my topic today is the Proteomics application with the Microtissues from InSphero. And before I show you two applications, I want to shortly introduce next generation proteomics to you and explain you what it actually is. Now, proteomics in general aims to quantify as many proteins as possible with high quantitative quality. So the analytical challenge in proteomics is that proteins are very delicate molecules. They are large and they're very heterogeneous in their physical-chemical properties. So the principle behind proteomics, high-content proteomics, is to first cut these large proteins into smaller pieces using proteases. They are like restriction enzymes on the protein level. They cut specifically at specific amino acids like lysine and arginine. So the result, if you treat a protein sample, a complex proteomic sample with these proteases, is that you get small peptides. So the peptides are much easier to handle. They are essentially small molecules with a similar chemical properties, so they can be treated all the same, and they are also very stable. So obviously, we are still interested in proteins, so these peptides serve as proxies for protein quantification. Now, proteomics using a mass spec works in a few steps, and it starts with the peptide samples, as I described before. These are purified peptides. For instance, from microtissue preparations, they are separated over a column. It's a HPLC column, just much smaller than what is usually used in a lab. It's a nano format. And as these peptides are sprayed out at the tip of the column, they are ionized and they enter the mass spec. So the main thing the mass spec does is to isolate each peptide mass or each ion at a specific mass. They are isolated, collected, and then they are smashed into pieces, fragmented. And these pieces are several amino acids, have specific masses, and what you get in the end, the real output, that's all what you get, are spectra. The spectra, you may have seen them before, are just the different masses with the different intensities. And this helps you for two things. First of all, as you can imagine, this is very information rich. It's like a fingerprint for each peptide. So if you know how a peptide fragment pattern should look like, you can easily match it. The second, the number of ions, tells you how much there is. So it provides really identity, very high specificity. I want to stress that the specificity is extremely high compared, for instance, to antibody-based methods where you never really know if it's cross-reactivity. Here you know exactly what you measure and you know how much there is. So in the end, it's about proteins, so we need to stitch together the single reads from the peptides into proteins. It's called protein appearance. But usually these peptides are very specific or very unique for certain proteins, so you can directly tell which protein you measured by measuring the peptides. So that's the basic principle, and it's the same for all the flavors of proteomics. Now, what's then the difference between next generation proteomics and classical proteomics? So next-gen proteomics is really focused on high content. It means many proteins measured from a single sample in parallel, and importantly, high quality quantification. And it's really based on a parallel processing versus sequential processing, which I will explain in a minute. And the hallmarks of next-gen proteomics is you can measure more, but more importantly, it's more reproducible and it's much more quantitative. And only this enables you to do the things that I will show you with InSphero microtissues. So classical shotgun proteomics works like I showed you before, in a sequential way. You isolate the peptides, you fragment them, you get a specific spectra. And then the next peptide is elute from the column, the mass spec, and so on and so on. So as you can imagine, if you have something like 10,000 expressed proteins and you cut it into smaller pieces, you have 100,000 of analytes. So it means at a given point in time, a lot of different peptides elute from the column. And it's virtually impossible to keep up with it in terms of speed. That means the classical shotgun is necessarily a random sampling approach. So you can only capture some of the peptides, but you will miss always some. And this missing is very random. Sometimes you measure it, sometimes you don't. And this has always been the main problem, that you get a lot of data, but it used to be very irreproducible. Now, the principle of next generation proteomics is quite simple. You don't isolate single peptides, but whatever elutes from the column at a certain point of time, you collect together and you fragment together. and you get the then convoluted mixed spectra of all the peptide ions that eluted at that point in time. So it's very complicated and certainly impossible to analyze by hand. But while we are living in the age of computers and with smart and advanced algorithms, it's actually very possible to deconvolute and to still say exactly which peptide eluted in which quantity at a certain point in time. And then you certainly do this over and over again over time, but it means whatever the instrument can potentially measure, it's recorded. You get complete data sets. Now in the publication with InSphero, so how good is this really? So our MCP publications compare this in a very systematic way. And if you look at the left, it's a very simple experiment. It's 24 injections of the same sample of a cell lysate. And here's the protein heat map sorted by intensities, subset of proteins. And what you would expect if you 24 times inject the same thing is that you hopefully get, well, some variance, but overall you want to see the same thing over and over again. And this is what you do with next-gen proteomics. You get almost complete data matrices. Compared to the data matrices you get with classical proteomics. I told you about the random sampling, and that's really an issue because it means you get all these gaps, these white space are gaps, where you can't really tell anything. So it's very hard to use that incomplete data matrices to really identify, for instance, regulated proteins. Secondly, you also see more because it's parallel and compared to sequential. But again, I think the more important feature is really the quality of the data. So now let's move to the applications. Two applications with InSphero's Microtissue. The first one is what Katrin showed before, the characterization of these three donor microtissues, but this time on the proteomic level. So you saw some of the functional assays from Katrin. So here the purpose is not to look at 4 or 5 different aspects, but ideally thousands of proteins. So you really want to know which among all the proteins in these liver microtissues that you can measure change. So the first thing you always do is generating a spectral library, and you can think of it as the fingerprint database that I was describing before. So we have an inventory, carefully evaluate which proteins there are in the sample, and then can use this over and over again. So this is what we did here. And overall, 3,500 proteins we found as, or qualified as measurable across all conditions. And the way we do it now is we prepare these microtissues and we measure each single one. And then we measure something like 3,420 proteins. And then we do t-test analysis for each protein to identify for each single protein if it changes over time or not. So in order to do this, we set the day 7 as a reference date. And we were looking at the abundance changes that there were at least twofold and statistically significant. And this is the data. Remember, it's 3,500 proteins. So on day 14, we found all in all 134 regulated proteins that slowly grows to something like a bit more than 300. So overall, it's less than 10% of all the proteins of the total proteome changes by a factor 2 or more, or also less. So as Katrin said before, I think overall, from a proteomic point of view, if you look at it, say, compared to the data we have from other projects where we have treatment conditions, it's a rather stable system. Now the second application, this is some of the data that Katrin showed before on the functional level. You can now nicely correspond to the protein level data. There's a lot of proteins we measure, but the beauty of the method is that it's not kind of a fingerprint method where you get just a global view. You can look still at each single individual protein as if it would be a single assay. Certainly in the cases where we can compare it, like with bufuralol, we know the protein that corresponds to it. We have here the first donor has a low level and correspondingly has also a low enzyme level. And here we have some slight changes over time in the second donor. You see it pretty well corresponding to the protein level. Now the second application is a classical drug treatment study. So here the analytical question is different. The question is which proteins change upon treatment of a drug with different doses. So it's a very relevant question. And typically, people would look at various aspects like a certain cytochrome P450, up or down regulation, phase 1, phase 2, phase 3 responses, single assays. Again, here we have the possibility, thanks to next-gen proteomics, to look at all the proteins at the same time. So the experiment was a 27 exposure, our exposure to various concentrations of acetaminophen, which is also known as paracetamol, in different concentrations from subtoxic to toxic, or only the highest was the toxic. And I want to point out that the analytical challenge here is really the small amount of samples. So these microtissues are certainly not very large, but we were able to work with 12,000 cells corresponding to one line in a 96 cell plate of microtissues, and to get a full proteomics data set from this. Now this is the experimental setup. We have 3 replicates each with different concentration of acetaminophen, control subtoxic condition IC50, and the toxic condition. And again, like the previous inventory, we made a spectral library, a protein inventory, proteins we deem to be measurable here, which is this time 2,800 proteins. And then we profiled simply all the samples over all replicates and identified the ones that were regulated. Now, so this plot shows you the analytical challenge again. So each line is a protein as it moves or changes over time. And as you see, it's really a needle in the haystack problem because there's so many proteins, and some of them change a bit, many of them don't. And if you imagine again the classical shotgun proteomics, if you have such a complex proteome and a large part of it are from technical point of view changing, then it's virtually impossible to really differentiate between the truly regulated proteins and the technical artifacts. That's why it's so important that next-gen proteomics really provides you with these complete data matrices. Now, overall, if you do our t-test here, we see something like 100 proteins expressed at the very low, changing at the very low concentrations already. Some of them up, some of them down. So later, it's about 50-50 going up and down, up to a bit more than 300 proteins change expression at the toxic level, but already at the IC50 level, we have 200 proteins changing. Now, again, as I said before, we know the identity of each of these proteins. So we can do things like looking at the classes of proteins that change. And you have here the different color codes, light to dark red, which correspond to different concentrations. And you see that some of the functional classes here, peaks R activation, for instance, already present at a very low concentration. Some of the functional classes like bile acid biosynthesis, glutathione biosynthesis are only becoming visible at the toxic concentration. And you can certainly do any other kind of functional annotation exactly as you could do with a microarray data set, because the structure of the data is very similar to what you would get from an RNA-seq or microarray data set, just that it's on the protein level, which in many aspects is the more interesting level. So another way to look at the data, again, at specific proteins that are associated to phase 1, phase 2, phase 3. Again, you see here different proteins in the color codes, different chips going up at already low concentration, some of them, and some of them at higher concentrations. Phase 3, we see more or see something at the highest toxic concentration. Phase 2, again, we have few proteins that don't change a lot at lower concentration, but some of them, like this UGT1A7, already shows a high level of regulation at the lowest concentration already. So another view of the data, which refers to the analytical quality again. This is something that the people used to proteomics may be familiar with. So the data in this raw data format looks like an MRM, multiple reaction monitoring data. That means it's very specific. And as you see, also very clean, which is a result of the processing by our algorithms. And you see the high quality of the data in the sense that you have a low signal here, then it's going up consistently over all the replicates. You can really have very high confidence that what you're looking at is exactly what you think you're looking at. And then you can also look at it as if it would be a Western blot, but just a Western blot with 3,500 proteins, and you can plot the protein profiles. Phase 2, another example, similar trend, upregulations over time, already a bit earlier here. In phase 3, very slight regulation, but again, because the method is so precise, you are able to statistically significantly pull out the regulation here, even though they are actually quite small. Now, finally, one of the interesting features of this technology is that because you measure everything that can be measured, you measure also things you don't know that you measure. So you can do really retrospective analysis. In this case, there's an interesting twist to the story. It has long been known that Acetaminophen causes certain adducts, NAPQI adducts, and it's known that they are occurring on cysteines. And in the lab, what people measure is the amount of cysteine modification. But you can just say that it's on the cysteine, not on which protein it is. So far, that means the specific proteins that are modified with this adduct have been unknown. So when we talked to our InSphero colleagues, we thought, okay, let's look at the data if we are able to actually find some of the adducts. First of all, associate them and see how they behave with the various concentrations. So what we did, we went into the highest toxic concentration, where it's most likely that the adducts are present there, and we actually could identify a few modified proteins. And these were 6 proteins, 4 of which are mitochondrial oxidative stress-related proteins. And then we developed these assays and went back into the data and profiled it. And what was very stunning to see was that already at rather low concentration, we found significant amounts of this modified peptide, in this case of PARC7. So nicely, we also could monitor the non-modified peptide. So in this case, we are even able to say something about the stoichiometry. So here, obviously, without acetaminophen treatment, everything is non-modified peptide, nothing is the adduct. But as soon as you start to apply it, you see that it's coming up here and then nicely going down here. So here is, I would say, 2/3 or 1/3 is modified, and toxic concentration more than 80% are modified. So almost quantitatively modified proteins with NAPQI adducts. And certainly, you have so many data points. You can also look at a completely different side. In the same protein, you see that the protein level doesn't change. It's certainly important to verify that it's not an effect of the protein level change, but really of the modification. So I want to summarize. I showed you how next generation proteomics enables you to reproducibly quantify thousands of proteins across large sample series, like time series or treatment series. And we can really get insights into the mechanisms of the proteome changes in these samples, the microtissues from InSphero. So we also use it in other projects with our customers. And overall, I would say that next-gen proteomics is really a powerful approach when it's about finding regulated proteins against the backdrop of the whole proteome, such as the biomarker discovery, mode of action study, or also identification of tissue-specific proteins. So finally, Biognosys is a company that has 2 product and service lines. So we offer what I showed you here, the example with InSphero's Microtissue reference lab services, where customers send their samples, can be various samples, and we do a protein profiling study, and we return the data, the regulated proteins and all the proteins, how they behave over time. This is contract research work that we do. But we also support our customers that do have their own mass specs in their facility with our products. And it's largely a software business, so it's foremost software tools, but also various reagent tools that enable to quantify and to calibrate these mass spec instruments. So I want to thank you, and I hope I could spark some interest in next generation proteomics, and we're certainly happy to get inquiries or any questions.
Thank you, Oliver. One quick thing I wanted to just clarify is that so each data point, each time point was generated, and for each concentration of acetaminophen treatment, that was done with 12 microtissues. Is that what you said? 1 row of a plate of microtissues, so you could get 8 replicates or 8 different doses on a 96-well plate of microtissues, and that's
correct,
enough.
Yeah.
Protein material suitable for analysis with the with the HRM-MS.
Yes, that's correct.
All right, very good. I just want to, I think that's important to let people know, exactly how many microtissues that is required. I think that's a very powerful tool for just 12,000 cells to be able to get that detailed proteomic analysis out of it, and that speaks to the sensitivity of your technology and also the metabolic robustness of the microtissues as well. So. Two very powerful technologies, I think, combining to give people a lot of, like you said, unbiased mechanistic, information out of a small amount of tissue. If you don't mind, I will take the presentation rights back and do a quick wrap up here. Thank you again, Oliver and Katrin as well. I wanted to let everyone know we will be posting this webinar for on-demand viewing in the coming weeks, and if you want to review that again, it will be available. Would also like to invite you to join InSphero for our next webinar, which will be next Thursday at 11 a.m. Eastern Standard Time. That would be 8 a.m. Pacific Time or 5 p.m. Central European Time. And that's a joint webinar being hosted by PerkinElmer, and InSphero's co-founder and Chief Scientific Officer, Dr. Jens Kelm, will present some recent work done with 3D liver microtissues investigating drug-induced cholestasis. And Dr. Karen Butcher of PerkinElmer will cover some really pretty cool high-content imaging work done with the Opera Phoenix high-content screening system to assess hepatobiliary efflux transport activity. And you can find more information about this webinar and register for that on the webinar page of the InSphero website. If you have further questions about today's webinar or specific questions for us about InSphero microtissues or to Biognosys about their next generation mass spec technology, we invite you to contact us directly at either sales or info@insphero.com or info@biognosys.ch. And if you're in the U.S., we'll be in La Jolla this week at the Applied Pharmaceutical Toxicology meeting. We'd love to meet you in person. And if you're in the UK area, or the UK is not far of a train ride or a plane trip for you, we invite you to join us at our UK user group meeting being held on Thursday, June 11th in Cambridge. So we'd love to see you there at either of those events if they're convenient for you. With that, I'm going to wrap up today's webinar. We thank you again for joining us. We thank both of our speakers for participating as well, Oliver and Katrin, and we will sign off and see you at our next webinar. Thank you.